This shows you that we can lose nucleosome-depleted regions, just to show you the power of this methodology comparing these two cell types. I won't go into this in any detail, but it allows us on the top to look at the accessibility, the exposure of the DNA, and at the bottom to look at the endogenous methylation. And what you see here is that regions which become methylated on a global scale also are the ones which lose accessibility. So, DNA methylation and nucleosomal positioning and histone modification are three parts of the epigenetic code that we're interested in. The future belongs to understanding how they interact with each other. Now, we've thought for a long time that this stuff was important in cancer. And I must say we met with a certain amount of resistance. However, what has happened over the last few years as a result of some of the things we've been hearing about in this meeting is that all of a sudden, there are a whole bunch of mutations which have been identified, which were completely unanticipated, which are in human cancers in genes which modify the epigenome. So, for example, we never knew about DNMT3A mutations at the top. Certain kinds of leukemia have lots of mutations there. And the ones that the histone modification enzymes are also frequently mutated. We knew about MLL, but the other ones are all new. And then on the other side with the nucleosomal remodeling, the machines are also very often mutated, or components of the machines. And so we never knew about these. 5 years ago, I would give this talk, there would be none of these mutations on there right at that time. So we're very excited at what has happened at the fusion of genetics and epigenetics. I want to briefly describe to you two of these things, what they do. A BRG1 is a machine, and ARID1A is a mutation, is a gene which is also in this remodeling complexes. These nanomachines are nano, but they're still pretty big molecularly, and they have many components. And for example, this here, BRG1 here, is a machine, as I'll show you, that moves nucleosomes around, and ARID1A, which is frequently mutated in cancer, actually tells the machine where to go. And so we've used our technology to try and figure out how these things might work. Collaborating with Bob Kingston, who is an expert in this field, we used our technique to show in a test tube how, if we made a nucleosome on top of a piece of DNA, it neatly sat right in the middle of the piece of DNA, but if we added BRG1 and ATP, the nucleosome in certain cases was moved to the left, and in other cases was moved to the right. As you can see, if you look carefully here, the position of the teal molecules has been changed. So this might switch a gene on, and this might switch a gene off. And so you can think of this as the machine needs direction as to what to do, and presumably the other components, which are also frequently mutated in human cancer, tell this machine whether to go forward or backwards to switch genes on or off. This just shows you the extent of involvement of ARID1A mutations in human cancers. Truly extraordinary, completely unpredicted, particularly in ovarian clear cell carcinoma. 50% of all ovarian clear cell carcinomas have mutations in ARID1A. So we tried, again, very early experiments, which have certainly not been published yet, try to figure out what does ARID1A do. First of all, we looked genome-wide using the ChIP-seq technology that, that you heard about earlier today to find out where does ARID1A bind on chromatin. And lo and behold, it binds very strongly and is preferentially located on CPG islands. That is regions of DNA which are often at the promoters of genes. So 42% of the binding is at these kinds of promoters and very few here. These are mostly housekeeping genes. These are mostly tissue-specific genes. And then you can do this kind of thing where you can try and figure out how they work. You can see that ARID1A is very neatly localized to the transcription start sites of these genes. Look here, for example. Here you have a peak of ARID1A binding, and we can knock it down here to try and figure out what its function is. This is in a gene called SMAD4, but just to show you, this gene is running in the opposite direction, E2F1. Again, this beautiful peak of ARID1A, which is bound there. And so you can imagine if we knock it down, we can ask, well, what a question can we simulate what would happen with a mutation? And we can do that. We found, again, I'm going to go through this very quickly. This is a gene which is a target for ARID1A. In this ovarian cancer cell line, the gene is completely silenced and has DNA methylation. You can see on the left, the pink that you're used to, that means it's silent. And when we knock down ARID1A, you can see that there are a whole bunch of alleles now that open up. Okay, so ARID1A is keeping that particular gene closed, and when ARID1A is not functioning properly, the gene begins to open up and begins to overexpress. So we will pursue this to try and figure out how genes are regulated and what the effect of these mutations might be. So we've figured out which genes are upregulated, which are downregulated. I just want to point out these inflammation-mediated genes are very frequently altered in these knockdowns. So I put up that classic drawing, which of course is wildly out of date, but this idea that information goes from DNA to RNA to protein. I think we have to remember that there is an intermediary step, and that step is called chromatin. That chromatin is deranged in cancer, and therefore the possibility exists that we can design drugs to target these defects. And in fact, the minister, when he introduced this meeting, talked about the Structural Genome Consortium, which is located, I think, in Toronto. A lot of their activity is focused on finding drugs which target chromatin. So chromatin is a hot target at the moment. And collaborating with Steve Balin at Johns Hopkins, we've been trying to push forward this idea that epigenetic therapy can be used to treat not only leukemias, but also solid cancers. And I want to finish with some results which are not my results, but the results from the team at Hopkins, which suggest that this may indeed be the case. So the idea is, and where we've had most of our success, is the idea of coupling epigenetic therapy together with immunotherapy. And why would we do that? Well, it turns out that cancer cells can evade the immune response by a process which is called tolerance, and that this tolerance is engineered by a series of molecules, including these ones at the bottom of the slide there, PDL1 and PD1. And drug companies have developed monoclonal antibodies to these two ligands to try and break tolerance and allow the immune system to kill the tumor cell. And what's been found with epigenetic therapy, we think that the main outcome of treating cells with DNA demethylating drugs and histone deacetylase inhibitors is to make the cell get inflamed, so to speak, and to become more visible to the immune system. And so what we found with some of the patients that have been put on epigenetic therapy trials and subsequently treated with immunotherapy against PD1, we get these amazing responses. And you can see this guy here, this is a lung cancer. And just look at the bottom there, that's the most obvious one. This guy's, I can't read it here properly, he's 61 years old. He has stage 4 refractory small cell lung cancer. He's had radiation therapy. He's had 2 multi-drug chemotherapy regimens. And then he was given 5-azacitidine and a tinostat. And you can see here this very remarkable site of this tumor actually really responding to this therapy. And so we're very enthusiastic about this and are pushing forward the idea that the future of this kind of therapy, of epigenetic therapy, may well be to combine it with standard other kinds of therapy. So I hope I've convinced you that cancer genetics and epigenetics are not two separate fields anymore, that these two fields do actually interact with each other. Epigenetics can silence genes, such as DNA repair genes, and cause genetic damage. Genes which modify the epigenome can become mutated, and this can alter the epigenome. So I think it's really important in the future to remember that these two things are actually linked closely together. Finally, I want to go back to this slide. Well, it's not, you haven't seen this slide before, but just to remind you that this word chromatin from Walter Fleming, this is a really bad malignancy. Just look at the nuclei, they're totally screwed up. They're all different from each other. You don't need to be a great pathologist to see there's something wrong here. So I'm just wondering if we're not at the cusp of finally understanding this after 120 years, that the reason why the chromatin becomes so disorganized is because many of the remodeling enzymes, many of the epigenetic modifiers are mutated in these cancers. And so maybe the chromatin doesn't know exactly how to organize itself, which gives us pleomorphic behavior. I want to thank everybody that collaborated with us. I particularly want to thank Steve Balin and the group at Johns Hopkins who did all the clinical work. That is definitely not my work, but I've been privileged to participate with them as part of a Stand Up to Cancer team. Thank you very much.
May we have some questions for Dr. Jones? Please, do you want to use this?
If this could be out, yeah. I'm astounded at the difference in these nucleosome-free regions between the epithelial cell and the cancer cell. I mean,
right.
I mean, it looks like 12,000 common sites, 40,000 different sites, and yet we know there's not that degree of change in the gene expression patterns between those cells. I mean, there is change, but it's not that extensive.
Yeah. So I think that's why I hesitated giving the slide. But we double-checked the data, and it looks right. I think there's two things that worry me. The one is that we're using this bizarre cell line that's been in culture for a long time, which might be related to that. And I think that many of the changes which pathologists see and which we see are actually could be structural changes, which is why I emphasize the change in the CTCF binding as an example. I think it's very early days, to say that there's a causal link at the moment, but I'm just very excited at the lack of commonality, and we're going to do this now with fresh uncultured tumors, and these structural changes and these mutations in these remodelers. I mean, it seems to me that if the remodeler is not working and a pathologist sees a bizarre nucleus, that there's a very good possibility that those two are linked. But this is very early days. That data is certainly just off the press. Well, not off the press. I wish it was.
Thank you, please.
I have two questions. I'm very thrilling to see like a small cell cancer of the lung can change so huge to be so clean later. And this is one of the most hardest cancer to be cured, and this is very stunning. And I have two questions. One is methylation usually only happens during CPG island, so you can turn on, turn off using epigenetics to control the gene expression. But other places beside the CPG island, other genes involved in cancer, is there anything in epigenetics you can give any suggestions? And another one is when you introduce about the anchor, the anchor into the chromatin, how they can help the chromosome to have the morphology. And you think it's because they incorporate more from your data. So I'm curious is if this is a self-defense appearance to try to stop, try to incorporate a lot of anchor and then try to stop the transcription. Mm-hmm. Or it's like it's a process initiation of the cancer.
Yeah. Well, again, I can't answer all your questions, but with respect to the first one, yes, CPG island genes which become abnormally silenced can be turned on by DNA demethylation therapy. That's clear. Whether patients respond because of that is not clear. And there certainly are, patients, there are genes which are not CPG islands which can also be upregulated. And so when we look and see what becomes upregulated after epigenetic therapy, what the predominant genes are genes of the interferon-like pathway, the STATs and the interferon-like pathway. So those are the ones that get most upregulated by mechanisms which are not clearly understood yet. With regard to CTCF, this often acts as an insulator between enhancers and promoters. And I'm just giving you a genome-wide analysis of what's going on there. We don't have any specific details yet, because I don't think we've started really zeroing in at that level yet. But certainly CTCF is often found at promoters. It is often found between a promoter and enhancer, and clearly can be involved in important regulatory processes such as imprinting.
Any other further questions? Good, thank you very much.